Good morning. I'd like to welcome everybody to day 2 of this workshop. I hope everyone who attended yesterday had a great first day. I know that I had a very good experience, a lot of useful discussions. So just wanted to give you a little bit of primer for day 2. So beginning the day, we're going to continue with our parallel sessions. The first session will be in vitro studies that may contribute to alternative bioequivalence approaches for locally acting orally inhaled drug products. And the presenters you see here will be giving talks on the topics of morphology and other in vitro approaches. The second session, which will be continuing from yesterday, will be in vivo and in silico studies that may contribute to alternative bioequivalence approaches for locally acting orally inhaled drug products. And the presenters you see here will be giving talks, and these talks will be about the use of modeling to inform PK metrics on regional deposition and regional deposition validation. And we're going to conclude the day with session 3. So the idea with session 3 is that we're going to take everything that we've talked about, coming back to the first symposium, which was about the comparative clinical endpoint and pharmacodynamic studies, and then session 1 and session 2, which are talking about various techniques that may be useful for an alternative bioequivalence approach. So more of the technical details. And then this third session is going to be a series of panels. It's really going to be 2 panels with more or less the same panelists in each panel. And we're going to be talking about how do we take all of these techniques, put them together into what might be a useful alternative bioequivalence approach. So you can see the presenters that we have here and the moderators that are going to be running these panel discussions. And these will be the panelists participating in the first panel. And after this, we'll have a break, and then there'll be a second panel with these panelists that you see listed on the screen here. And concluding the workshop, we're going to have closing remarks from Dr. Bing Li. And that will conclude our workshop. So thanks again for coming today, and I hope you have a great second day.
All right, good morning and welcome for this session 1's day 2 discussion. So we'll walk through the presenters that'll be giving presentations this morning. So I'll go through some of the introductions for these. So starting off, the first presenter will be Dr. Nicholas Holtgrewe. So Dr. Nicholas Holtgrewe has been a chemist at the FDA Division of Complex Drug Analysis, DCDA, within CDER OPQ OTR since April 2019. His research at FDA focuses on inhalation drug characterization, particle sizing, X-ray powder diffraction, and spectroscopy, and his expertise is in optics, X-ray diffraction, and Raman spectroscopy. He received his BS degree in chemistry from Truman State University in 2008 and a PhD in chemistry from Washington University in St. Louis in 2013.
Thank you for the introduction, Bryan. Our second speaker is Dr. Shur. He's an academic and entrepreneur, having over 100 published scientific papers, and was CEO of Nanopharm, which he led to a successful exit in 2019. JAG's main area of research and commercial endeavor is in the complex topical pharmaceutical drug products area. A key theme of his research and commercial enterprises has been to develop patient-centric drug development platforms. He began his career with Profile Drug Delivery, now Philips Respironics, where he developed a liquid dose drug delivery systems for cystic fibrosis patients, and later went on to work for GSK. Jag holds a bachelor's in chemistry and completed his PhD entitled Formulated Mucoregulatory Agents in the Airways of Patients with Cystic Fibrosis. Following this, he was a postdoctoral fellow at the London School of Pharmacy, investigating the fabrication of microparticles for vaccine delivery using supercritical fluid technology. Jag remains a part-time member of Nanopharm, an Aptar pharma company, as Vice President of Science and Technology. Our third speaker will be Agisilaos Kourmatzis. He is an associate professor at the School of Aerospace, Mechanical, and Mechatronic Engineering at the University of Sydney, Australia. His main research focus is on turbulent two-phase flows, with a particular interest being the development of optical and laser diagnostic methods for the improved characterization of aerosol dynamics. Relevant to inhalers, he has recently focused on providing more detailed insights into the mechanisms of particle transport within DPIs and physiological, accurate replicas of the human airway. Mechanisms of deagglomeration, deposition, and particle gas mixing are of particular interest. Dr. Kourmatzis has a PhD in fluid dynamics from the University of Southampton, UK, and a master's in advanced mechanical engineering from Imperial College London, UK, and a bachelor's in mechanical engineering from the University of Nottingham, UK. He's a professional chartered engineer. His research has been supported by both industry and government, including Australian Research Council, Mitsubishi Heavy Industries, Proveris Scientific Corporation, the US FDA, Sishu Singmed, sorry for the mispronunciation, the Great Barrier Reef Foundation, Venzo Labs, and others. Our fourth speaker will be Xian-Ming Zeng. He holds a bachelor's and master's in pharmaceutics from West China University of Medical Science. He obtained his PhD in pharmaceutical science in 1997 with a focus on particle engineering and dry powder aerosols from King's College, London, UK, where he was subsequently appointed the Maplethorpe postdoctoral fellow. Dr. Zeng joined IVAX in the UK in 1999 as head of pulmonary research London, leading the development of several inhalation products. In 2005, Dr. Zeng became professor of pharmaceutics, head of Department of Pharmaceutics, and director of research at Medway School of Pharmacy, University of Kent. In 2007, Dr. Zeng relocated to the US to serve as head of respiratory product development to lead global respiratory product development of Teva branded pharmaceuticals R&D. In 2013, Dr. Zeng joined Lupin to establish their inhalation development and manufacturing capabilities in the US and lead their inhalation development activities across US and India. He was serving as executive vice president and global head of inhalation and complex injectables R&D when he left Lupin in late 2021. Dr. Zeng has since founded Transpire Bio Inc. in South Florida, dedicated to the development of generic and innovative inhalation products for advanced markets. Dr. Zeng has over 25 years of combined experience in inhalation academic research and product development, authored 50+ peer-reviewed publications, and led the development of a number of generic and innovation products marketed in the US, UK, and EU. So the title of my talk today is Alternative Bioequivalence Approach Using MDRS, Morphologically Directed Raman Spectroscopy on Nasal Spray Suspensions. So before we begin, I realize that nasal spray suspensions are not orally inhaled drug products, but they are very similar in that they are locally acting, and I think we can learn a lot from a success story of using MDRS for product-specific guidances. And this is to kind of bypass this endpoint clinical study. Since I work for OPQ, a quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It is what gives patients confidence in their next dose of medicine. So for my outline today, I figured I'd go through just a brief introduction to what MDRS is, how it operates, how we choose, which morphological characteristics to kind of hone in on, and then I'll run you through an example of a success story of how we use MDRS to bypass these, as we learned in previous talks, expensive endpoint clinical studies. So why would we want to do this to begin with? Well, if you learn one thing from this talk, it's that we are after the particle size distribution of the API in the drug product. So we want to isolate this from everything else that's in the formulation. So that is what we are after. This assures we get quality, effectiveness, and what we are ultimately after is this bioequivalence for evaluating generic drugs. And that's why we're here today. Some of the challenges that MDRS addresses. So if you have excipients in the formulation or any other types of particles, you can isolate the API from the excipients if they coexist. You can isolate more than one API. I've seen up to 3 APIs in a formulation before, and MDRS can address this. And then if your API has more than one polymorphic form, depending on the drug, the API may be able to help you out here because different vibrational modes can isolate different polymorphs. So traditional particle sizing techniques, cascade impactors, laser diffraction, and just microscopy alone, they're kind of blind to these APIs. They just assume everything is either API or non-API. So this addresses those issues. So a brief introduction to MDRS. Put simply, it is just the combination of microscopy, which I'm sure we're all at least a little bit familiar with. You just look at your sample under a microscope, and then you combined it with an ID portion. So this uses Raman spectroscopy in this case. The microscopy, you can see on the bottom left, here's an example image of what you would find if you put a nasal spray suspension under the microscope. So you can see, there's tons of different shaped particles, elongated ones, short round ones, dark ones, light ones. You're not really sure what everything is. This is where the Raman spectroscopy comes in. And you can actually shine the laser right through the objective lens using a confocal arrangement. And the way Raman spectroscopy works is you start with an excitation laser with a known narrow frequency, and you shine that on your sample. Now, what happens is the sample undergoes molecular vibration, and this can absorb a very small amount of that excitation laser frequency. And when it emits, so that scattering effect, you effectively get a photon with less energy than what you started with, the excitation laser. You pass this through a bunch of filters. Like a very, very small amount of light is Raman scattering. It's not much. So you have to have very sensitive detectors for this. So you pass it through a bunch of filters, use a diffraction grating to spread out the light that you want to detect. And what you end up with is a spectrum at the bottom right-hand corner where the x-axis is the Raman shift. So this is that lower frequency range that I was talking about. And 0 on this scale is the excitation laser frequency. And what you get is a fingerprint of, you can kind of isolate different peaks and different intensities of different vibrational modes of your drug product. So everything has kind of a unique signature. And so you can pick out API and excipient. So morphological characterization and screening. How do we kind of, everything's a different shape, everything's a different size. How do we unify this? And the way that we want to look at the data is with this circle equivalent diameter. So on the top right corner, you can see I have a picture of a 3D particle. So this is kind of our microscope image. You capture that image as a 2D image, so you pixelate it. With those pixels, you can calculate an area and convert that into a circle, and then measure the diameter of that circle. So everything's now unified. So when I speak about circle equivalent diameter, CE diameter, this is what I'm referring to. Next is, I've just given here 2 examples of morphology characterization. You can go, there's a lot of different ways to analyze this data, but I just picked out 2 just as an example: aspect ratio and circularity. So, for aspect ratio, you can see you can have elongated particles, you can have short square ones, and then for circularity, you can have how close is that particle to a perfect circle is kind of what that numerical value represents. So when we talk about nasal spray suspensions, I want you to think about MDI suspensions. They're very, very similar in the sense that establishing bioequivalence for locally acting drugs is challenging. Traditional PK studies, they do not apply here for locally acting drugs. And so when we go test this, we want to make sure that the device is included. So for nasal sprays, we use sprays directly from the nozzle, and when you use, for example, MDI in suspension, you would activate it from the device itself. So I'm sure we're kind of all sick of seeing the weight of evidence approach, but I won't spend too much on this slide, but just to really drive this home, this is where MDRS lies in this diagram. So we want to try to avoid these endpoint clinical studies. So jumping into how we collect samples, and I will run you through this brief example of using nasal suspensions as an example. So we use a five-step method. It's debatable whether the fifth step is something that is definitely something that you have to do, but we'll do 4 or 5-step method, sure. So we start with a sample preparation. We will go into how we image and how we analyze the morphology characteristics, and then what parameters we use for Raman measurements, how we classify things as API or non-API, and then we'll jump into morphology filter selection. And then lastly, optional, we will determine the minimum number of particles for detection. So when you first start an MDRS experiment, one thing that you want to do definitely is have a training set. And this will be time consuming, but it will be worth it, because remember, what we're after is the API particle size distribution. So initially what you want to do is you want to collect as big of a training set as you can. In this case, we're collecting about 10,000 randomly selected particles. And this will include API, it'll include excipient, it'll include anything else that's in the formulation. And this is what we want to use to gain an understanding. So you want to understand your sample, you want to understand your formulation initially. So for sample preparation, since this is a suspension, we will be using a wet dispersion method. And dry dispersion, so if you just let the drop sit on a glass slide and let it evaporate, you're going to get a lot of agglomerates. It's going to be a mess. You're going to have particles touching each other. That's not to say that you can't use this on DPIs. There is a separate instrument that allows you to load powder and have a puff of air disperse that powder and allow it to settle onto a glass slide. And that works really well for separating out particles onto a glass slide. So for the wet dispersion method, what we do for the nasal spray is we prime the nasal spray, 2 actuations, and then we collect 2 actuations into a glass vial. Immediately pipette about 5 microliters onto a quartz microscope slide and cover it with a quartz cover slip. So that's kind of the image that you see in the lower right-hand corner. We seal this with petroleum jelly, and that prevents evaporation. And this works really, really well. A sample is allowed to rest for about an hour. This is kind of, formulation dependent. Really what you want to do here is look at your sample under a microscope. If you see browning in motion, then let it sit longer. That is the ultimate goal. We do not want these particles moving around while we are analyzing the sample. So for some of the details that are specific to nasal spray suspensions, but could also work for MDI suspensions, we will start with a scan of radius 2 millimeters. So I just have kind of an example of the size of the cover slip in the scan area in the top right corner. We're going to collect a minimum of 10,000 particles. We use a 50x objective lens, and I know we said we're not going to filter anything initially, but we do want to filter anything less than 1 micron because the Raman laser spot size is this is about the limit at which you can detect things. So we'll filter anything less than 1 micron circle equivalent diameter. The light intensity is important to be calibrated if you use an intensity mean as like a morphology characteristic. The edges of the particles, they will be determined by image binarization. Now, there are really good algorithms nowadays, and this is a part where AI could even come in and pick out specific particles for you. In this example, we are using a simple binarization shown on the right-hand side here, where you just pick a threshold value, and if your particles have good contrast with the background, then it picks it up really easily. Lastly, the software that we're using logs the XY coordinates. You can actually go back and check for Raman ID. So when you run your sample, this is kind of like what you would just pick out a few particles just to look at, randomly selected. So as you can see, part of this formulation has small round dark particles, others that are transparent and elongated, and then you have larger square type particles, and then the particle you see on the far right-hand side is actually 2 particles touching one another. So we want to try to avoid this as much as possible. Even though you can kind of like push these out with filters, it's best to try to avoid them to get the most accurate depiction of your sample. So now we come to the ID portion of this. Remember, we have no idea what those things were on the last slide. So the way the software works, it goes back to those XY coordinates and collects a spectrum, as you see on the left-hand side here, and we can kind of use a reference standard for the API that you're working with and compare that to the spectrum that you've collected. So in this case, the one that has the 4 peaks that are around 1600 Raman shift, that is the mometazone furorate, and then the one that has the peak at about 1100 wave numbers is the microcrystalline cellulose. So we can see now we have an ID, we have the picture. Now we can start to really develop a picture of what's going on in the formulation. But I must admit that this is a these are very good spectra. Not everything is this pretty. So surface roughness can affect Raman scattering efficiency. So on the right-hand side here, I show spectra that correlate to different API like correlation values. So what the program does is it will compare that to the reference spectra and assign a value between 0 and 1. 0 meaning it's nothing like the API, and 1 being it's a perfect replica of the API. And so you have to have a cutoff. There's a continuum here. So what we came up with was for mometazone, we said at least one of the 4 API signature peaks had to be present. And this intensity had to be acceptable within greater than 3 signal to noise ratio. So that was our criteria, and it ended up being that the spectra in the top left corner would not be labeled as API, and then the other 3 would. So we have our training set. It consisted closer to 11,000 particles. And then of those 11,000 particles, we identified about 12% of them as API particles. And I put a disclaimer here. This is just dependent on this formulation in particular. Every other formulation will vary from this value. And we found that a typical excipient particle was elongated and transparent, shown on the right, and a typical API particle was close to round or square-shaped and opaque. So we have from the training set, we have our API particle size distribution. Now, you could just say, okay, we're done. That's what we wanted. That's what we came here for. But the downside is this equates to about 90 hours of collection time at 20 seconds per Raman collection. So we kind of want to shorten this. Again, you don't have to. If you want to shoot, you shoot particles for ID, and you could just hit, excipients the whole time. I mean, go for it. You'll still get an API particle size distribution, but you can reduce the time significantly by using filter selection. So when you choose filters, what you want to do is you want to make sure to not remove as many, like you want to try to keep your API particle size distribution that you got from the training set consistent. So it must match that. If you cut it off too much, then you're going to affect this. So when we look at our data, you can see we've plotted the excipient and API data from using different morphological characterizations. So we have, circle equivalent diameter, aspect ratio, intensity mean. And you can kind of visually see that there's differences. Where we pick the filter is kind of debatable, but you can kind of qualitatively see, okay, aspect ratio, I'm going to choose about 0.6 to cut off as many excipient particles as I want and retain a lot of the API. So when you pick filters, this is what you want to keep in mind. And try not to pick too many filters, but it's a messy process, I will admit. But ultimately what we're after is you want this API particle size distribution, and you want to extract this D10, D50, and D90 from it. So not to throw a bunch of numbers at you, but this is a multi-dimensional problem. I mean, I showed 5 examples of how you can characterize particles on the last slide, and there are more than that. So we chose to use aspect ratio and intensity mean as a means of cutting out all of the excipient particles, or as many as we can, and then retaining that API particles. So we ultimately kind of you end up iterating, like this is what the columns are doing, is you start with aspect ratio of 0.6, intensity mean of less than 150, and then you iterate until you get to like a maximum. And you want to try to retain as many API particles and remove as many excipients as possible. And what we're monitoring at the bottom is how far does this vary from the original training set API PSD. So we would try to limit that variation to no more than 3%. For the D10, D50, and D90. Once you have that, you can apply your filters. Then the next time you go and scan your sample, you're going to be only IDing, or majority of the particles should be API. And so as the last thing, you can determine kind of like number of particles. What we were trying to do here is kind of mimic in vitro characterization where you just randomly select. So this is just straight from the training data set where you pick, you run 5 example runs, but the criteria, we just kind of wanted to see where we've plateaued at a steady state. And this should, eventually plateau to where the training data set, that gold standard of API particle size distribution, what we want. And we determined that for this sample, you needed about 400 API particles to accurately characterize the particle size distribution. And again, this is debatable. It's kind of a bootstrapping method, but it kind of gives us a little bit more confidence in the filter selection. So in review, we covered MDRS, how it combines with Raman spectroscopy, and how we can use it for establishing bioequivalence in generic nasal spray suspensions. So just get those gears turning for MDI or DPI formulations. Then we covered the example for a nasal spray suspension where we went for sample preparation. We did the wet dispersion method. For particle imaging and morphology analysis, we determined we got these transparent and non-transparent square-shaped particles. And then Raman ID'd those particles. And when we classified everything into API excipient, one little gripe here, and this is just from my background, is when you identify something, if you do not, if the spectrum is flat, don't ID it. It's labeled that as unclassified because if you don't, pick up API and you label it as excipient, that can become an issue later on down the road. We also covered morphology filter selection. So what we ended up with was an aspect ratio of greater than 0.6 and an intensity mean of less than 160 to isolate the API from the excipient. And then we determined the minimum number of particles to be about 400. So just kind of my last two slides are just to show you that, product-specific guidances, nearly all nasal spray suspension products have this in their language. So MDRS is very popular for these formulations, and I don't see why it couldn't happen for MDI suspensions as well. And just to give you like a list of some of the nasal spray suspension products. And then lastly, these are the two papers from the FDA that I've been kind of, talking about this entire talk. So if you'd like to learn more, I've listed them here, and I believe the slides are available on the website. And just as kind of like a final note, I would like to say that, ultimately what we were after, that goal of getting the API particle size distribution and isolating it from your formulation, it doesn't have to use MDRS. Like, you can use spectroscopic methods that are, non-destructive to your sample. So don't feel like you're limited to the RS portion of this. You just need to ID your sample. So yeah, thank you for listening.
Well, good morning, everybody. And well, firstly, thank you to my FDA colleagues for inviting me out for our first face-to-face. It's been a long time since we've been fortunate enough to actually talk about these things in person. I think yesterday it was evident that there's still so much that we have missed in having these open discussions. Now, I was asked to talk about morphology, and my background is all single particle. I've worked in measuring forces of particles for most of my academic career. So I look at things more from ordered and structure. So the way I look at pharmaceutical products holistically, they are structured systems. But when we come into the respiratory area, whether it be nasal or for pulmonary delivery, we're dealing with systems which are structured at the micro scale. So the important parameter that we need to really start focusing on is this concept of microstructure. Now, what we have with respect to formulation systems which are created for aerosol systems, we had some talks talking about the ADVEP DPI. We've had discussion this morning from Nicholas on nasal suspension, nasal sprays. Ultimately, there are some of the properties of the raw materials that are incorporated into the formulation, how we process them, and how that allows the formulation piece to structure together. So this structure is formed at the point of formulation manufacture, and it's going to be controlled by the surface interfacial properties and the particle size and the surface morphology of the raw materials that we're going to utilize. And that ultimately gives us a performance. But for a long while, if we take Nicholas's example for nasal sprays, when it came to showing equivalence for nasal spray suspensions, we had not just a variable pharmacodynamic study. We had a pharmacodynamic study which needed to be timed for seasonal, at a specific moment in time in the season. And therefore, when it really became prohibitive for allowing generic products, particularly for nasal suspensions, to come through. The introduction of MDRS has really changed that. I think it's been a huge success, and is, as you can see, that there's 6 product-specific guidances specifically for nasal sprays which incorporate MDRS, and it's been I think has unlocked the potential for the nasal spray area. Now, what the FDA, in terms of how the 505J statute is written, it talks about having scientifically valid measurements, so scientific tools that we can validate, which allow us to understand the local rate and extent of absorption. And that primarily intertwines with the use of MDRS, measure the particle size of the drug in the nasal suspension. It's going to correlate to its dissolution and therefore its distribution and absorption from the nasal cavity. Now, the step for inhalation is slightly different. because unlike with nasal sprays where the drug deposits wherever it lands in the nose, so it doesn't need to traverse through the throat in order to enter the lung. Wherever the dose goes in the nose, it's going to deposit, and these effects will take effect. So we have to build an arsenal of tools which actually allow us to understand the microstructure of the aerosol system. And there are aspects of MDRS and other tools we can use to understand that microstructure. And what we postulated from a research perspective, and what we've positioned over the years together with the agency, is this introduction of a Q3 tool set. So this idea of understanding the state of matter and aggregation of these systems, and allowing that to be a way of, at least from an in vitro perspective, to understand the product. So when we started to explore this area, it was to really use these tools to understand the reference product, and then you start driving forward the formulation development piece for your generic product. But it's our position was always to really look at developing Q3 tools which allow us to look at this particular area. Now, Nicholas has already gone through this. We've used MDRS for nasal sprays, and this is sort of a very similar approach, and it does rely on sample preparation is key. You're trying to catch the drug in its native state in that formulation system. And we have a similar sample preparation to how Nicholas described it. We then introduce those filters to actually look at those particles and identify them and be able to contrast them in this microscopic tool, apply a series of morphological filters. So now we're looking to remove the solid excipient particles out of the count so that we're biasing everything around the drug, which we're interested in. And then we start doing the chemical analysis. And I think it's been an amazing tool in order to allow us to really explore the particle size variance, not only for the reference product, but also for the test product as we start developing them. So Nicholas mentioned the use of aspect ratio, which I've termed here as elongation, and its utility as a way. This is the primary filter that we're using to remove those particles. So everything to the right, so the red curve, the blue and the green, essentially are excipient particles to the right. The big yellow is just the yellow side where you've got a smaller elongation is all drug. So you can put your morphological filter in that green zone to essentially cancel or remove those elongated particles. And the way we've looked at introducing those filters is to look at how, at what point does that circular equivalent diameter change. So you pick a filter where you're seeing the least variance in the size that you're trying to measure, and then you're away. But one point I'll make to those interested in the nasal area, when you just look at this plot, it's not just important to look at the drug. Actually, the quality of the excipient is also important. And actually, if you just look at particularly the red and the green, the red and the blue curves, those are just placebo. But you can start to see there is some variance in terms of the morphological structure. And from a device formulation interaction, it does have an influence on the formulation rheology. Now, when we started to explore this, and we've published a paper with Elizabeth and Bryan looking at trying to track particle size measured outside of the formulation. So you buy formulation in, and here we've got a series of batches with different particle size. Primarily batch 2, the red line, has a smaller particle size, and you have the batch 3 and 4, which have batch 1, which has a larger particle size. And when we formulate and then track the APSD through MDRS from the nasal sprays, there's a good correlation. We can track the particle size that we've brought in and as received, and when we put it into the formulation, we're able to track that, with the exception of formulation or batch 4, where you can start to see that actually the formulation process itself is dispersing the API and actually is a lot closer to the reference product in that behavior. So the paper that we've published actually talks through a lot of the challenges in terms of selecting a number of particles. And I think Nicholas mentioned bootstrap analysis, which is a very statistically sound approach of looking at particle number that you're going to use in developing a method. And that particle count is so important, particularly when you start to move away from using MDRS for other drug products such as DPIs and nasal suspensions. Another consideration, this is a paper that we've the paper on the right, understanding the number of containers that you're going to use for your IVBE study. How do you power the study, and how many sectors through life of the container are you going to use for demonstrating that IVBE? And that is where the burden of testing starts to ramp up. So whilst this is a great methodology, there needs to be a simulation of the number of test product, number of reference products that you're going to use through life sector, how you're going to assess this measurement, because it does have an impact on the downstream, some of the cons or some of the disadvantages of this system, which I'll talk about in a minute. Now, I've never just been an advocate of MDRS. I think you need to use orthogonal methods. We use that term quite broadly, but we always felt, well, if I'm measuring particle size, clearly dissolution is going to be an important measurement for understanding that correlation in terms of particle size and the effects on dissolution. And here, we're not trying to use dissolution as an IVC tool. We're using dissolution just to look at differences in how these different batches dissolve. And if you look on the left, the cumulative mass of these different batches made with different particle size are enormously different. And there is and we actually model the dissolution half-life, which is the plot on the right, and compare that and plot that against the volume distribution from MDRS, so the percentage less than 5 micron. There's a very good correlation. And I think this is really important because, one, it actually provides further validity to the MDRS method. And you can start seeing how you can actually track the quality of the product through your API particle size from the point of you buying your drug in to the point of you formulating, and then as you start moving into the IVB. So you're building this into the development much, much earlier. Now, the other importance of using dissolution alongside this is MDRS is, there are a few issues that we need to consider. One is that there's a limit in terms of how small, the small particle fraction. We really will struggle to see the particles below maybe 1.5 micron. So that's the limit at which it can really, really be pushed. So then you're always going to have this nascent question about what's happened to the small particle fraction. What's that small particle? They're there, we know they're there, and they will have an effect. The other thing which I just want to highlight is that these runs are long. And what we've noticed is there are system reliability issues, particularly with laser failing. Nicholas mentioned the XY tracking, sometimes that fails. So there are robustness issues, which I think as this is going to be used further in the future, there is a point for us as a community who are using this to really address some of these things. And that's something I'll leave as a point for discussion. So, but I don't want to stop on a negative. I think this has been a hugely successful venture, and in terms of allowing, and it's opened up the nasal spray generic area, and it's allowed a lot of understanding to come in for the new products for nasal sprays. Now, when we, Nicholas mentioned, well, let's go away and let's start looking at using this for orally inhaled drug products. But there's a bit of I mentioned one of the problems. One of the problems is that we are dealing with an aerosolized dose that has to traverse the throat before it actually gets into the available, to be total lung dose available for absorption from the airways. Now, yesterday we spoke about realistic APST tests and the use of anatomical throats, and they're a fascinating tool because actually they allow us to look at the realistic airflow path that materials will take once they're aerosolized. But there are other dynamic processes that are occurring in different types of orally inhaled drug product dosage forms. I'm not going to go into MDIs today because I think I'll be here for another hour talking about them, but I will just mention about dry powders and focus a little bit on them as well as a little bit on SMIs. Because even if you take a soft mist inhaler and you think, well, I'm just making a solution aerosol droplet, the droplet dynamics from an evaporation and condensation perspective in the airways, it creates its own type of structure. And that has some interesting facets when you start understanding, well, how do those how does particle size change and therefore impact regional deposition. With DPIs, we are always fighting the cohesive adhesive balance of these systems. We're trying to overcome the cohesion of the drug whilst maintaining flowability. And as a result of that, we end up creating a formulation microstructure. And in essence, we started with, if I just move on with the DPI story, Advair was a great learning curve for all of us. And if you just look at the PK, this is just something we've taken from the literature. And what we've done, what this plot is trying to do is compare the PK exposure from two doses of 150, so the low dose Advair product, with one shot of the mid dose, so roughly equivalent. Now you can see from the PK, the PK exposure, it's different between two products which have a nominally the same dose. So the first thing when we're trying to build Q3 tools for orally inhaled products, the first thing we've got to all believe is, and the data is there, is PK is a very, very sensitive tool for understanding differences in regional deposition and how things deposit in the airways. Now, what we've tried to do in the past is link PK directly to in vitro measurements such as cascade impactors. So on the right, what I have is just a NGI plot, not only of the fluticasone salmeterol components for the 150 low dose product of Advair, but also the lactose. And what you see is that the API is never alone, right? Even it's there in the presence of quite a significant amount of lactose. And what we were interested in trying to understand is, well, what does that structure actually look like? Do you have individual particles of lactose and agglomerated particles of drug? Is that the missing link in terms of understanding the exposure differences we're seeing in these formulation systems? So in order to do this, what we had developed is an aerosol dose collector system, and we've published this together with the agency. And what we're trying to do here, and what has been sort of a difficulty in this area, has been trying to collect the aerosol dose in its native agglomerated structure. So when we try to do this in the cascade impactor, the jets of the impactor basically piles everything up, as Gunther mentioned yesterday. And essentially, it gives us an erroneous agglomeration that doesn't represent the product. So this aerosol dose collection unit, we validated which it allowed us to catch the aerosol dose as the aerosols created it, as the aerosolization processes created it. And that allowed us to, one, collect the dose and to conduct dissolution studies to understand the particle size effects and the morphology differences, but then to also tie that in with MDRS. So we can take the same cassette with the particles deposited, put it under the MDRS and put it into the dissolution studies. And this is something that we found when we were really interested in comparing the European and the US versions of Advair. And what we found when we just looked at the dissolution of the FP component, well, firstly, there's an orange dot, which is representative of the Flixotide or flow event DPI at the same low dose. And the FP dissolution is a lot slower than it is in Advair or Seretide. But importantly, Advair's dissolution, the FP component of Advair, was much faster than that in Seretide. And when we looked at the microstructures using MDRS, which is the plot on the right, so here we're looking at not particle size, but just what fractions of agglomerates do we have. So the light blue boxes are just free FP, and then the orange boxes are FP agglomerated with lactose, and then we've got the yellow boxes and the darker blue, which are FP salmeterol lactose. The first thing we noticed was that there's not much freestanding FP. FP is a poorly water-soluble drug. It's poorly wettable as well. So having more freestanding FP makes it difficult to dissolve because it's going to take time for it to wet. Whereas, which is what we see in Seretide, it's a lot more freestanding. And what we also see with the Advair product is there's a lot more FP lactose interaction. And what we started to see was perhaps this is what's controlling the behavior of these two products. And when we did some multivariate analysis of the differences between Advair and Seretide, and primarily what we saw was Advair was more characterized by these FP lactose agglomerated systems, while Seretide was characterized with more of this FP salmeterol. We knew from our single particle studies that FP will always adhere to both lactose and salmeterol. But here, for a European and US product, you have differences. Now, these differences could be attributed to the raw materials, the process, where they're made, but importantly, they do have characteristically different dissolution behaviors for the FP component. Now, to take this one step further, we were working with the agency in running this dry powder PK study, where we took a single batch of FP and tried to modulate its APSD so that we can impact it or change its regional deposition. Now, we had done, talking about AI, we had used some AI models from data from our own lab just to look at what do lactose fines actually do to DPI formulations. And what we found was if you use microfine lactose, more than likely you impact the fine particle mass. If you start including more of the larger lactose particles, so between greater than 15 micron but less than 30 micron, they allow us to shift MMAD. So these formulations were designed to start really primarily, and Gunther mentioned this yesterday, we didn't really want to change structure. All we wanted to do was change where it went. And we had no idea that there will be a structural effect. We could probably hypothesize it, but we weren't sure of it. So here what we ended up with is formulation A, which had a much larger MMAD than formulation B and C. Formulation B and C was more milled micronized lactose. Formulation A was a much coarser milled lactose that we had incorporated into the system. Now, the bottom left is the PK and it looks very similar to the example of the FP100, FP250 I showed earlier. So formulations B and C are flying in terms of the exposure in the lung. Formulation A is taking much longer to get there. In fact, the Cmax are not comparable. Now, if you look at the plot above that, that is just the aerosolized dissolution on the aerosol dose collector. So that's collect the dose, put it on a USP2 paddle over disc experiment. And what you see is that the dissolution of the FP components faster in the A and B formulation, the B and C formulation than the A. Question is, well, what's happening to the microstructure? And again, what you see is that with formulation A, the slowest dissolution, more freestanding FP, and B and C have actually far less freestanding FP and more of it actually agglomerated with the lactose, which is aiding that. So that change in structure is allowing us to modulate the exposure and the release of the FP from the system. And I think this sort of enables us to start piecing together, well, what have we learned from this process? That we can use these tools to understand how we process formulations, how we change raw materials, how they come together to actually give us not just a cascade impactor, but a further set of data set, which allows us to look at that structure and how we can modulate that performance and correlate that back to PK. And the importance of this is, if we ignore the PD and the clinical endpoint for 1 minute, if we can make formulation changes in the test product that can modulate the PK, and we can measure the in vitro parameters such as MDRS, such as dissolution, and maybe others that can show that correlation, then I think we are in a place where we can start developing some of these Q3 tools. Now, I'm going to just talk a little bit about an area that I'm working on as a hobby at the moment. I'm really interested in dynamic aerosols. I like understanding, and I think you're going to hear from Aggie later about how that plays out in DPIs. But I'm really interested, going back to where I started, which is in nebulizers and soft mist, and I'm trying to look at this for MDIs also. So I'm collaborating with a spin-out from the University of Bristol Aerosol Science Group and a company called Microsol. And what they have is basically a single aerosol capturing system. So you have an aerosol dispenser, you put your formulation into it, it disperses an aerosol, and it catches a single aerosol droplet. And what you can do then is actually measure its size and change in morphology as you change humidity and as you change the environmental parameters around the system. So the data I'm showing here is on the y-axis on the right there, you have the radial growth factor, which is basically how much a droplet of a drug increases in size as the water activity or the humidity increases, which is on the x-axis. The take-home message is, if you just compare salbutamol, tiotropium, ipratropium, the radial growth factor of salbutamol is significantly higher than tiotropium and ipratropium. In fact, what that means is that once it goes into a high humidity environment, i.e. your lung, it's going to undergo a much rapid change in terms of aerosol growth. Interestingly, tiotropium has a faster radial growth than ipratropium. And you can start postulating whether this is a chemistry, what's actually driving this. But here you have a basis to show, well, actually, when these droplets are exposed to these conditions, how the hygroscopic growth is going to be different. So therefore, the morphology, the particle size, the structure of the aerosol, this is just a simple aerosol system, you think. And interestingly, as you start to then add excipients, this sort of evolves. But I'm just showing this as just an interesting point that it's different between these molecules. Now, when you start to then model that particle size distribution using this apparatus, the first thing we want to point out is it's a bimodal distribution. That's the first take home that we need to sort of just look at this. And so you have a bunch of small particles and a bunch of large particles in that sort of 10 micron area. And this is universally true for soft mist inhalers. It's always a bimodal distribution. And it's really difficult to capture this under standard cascade impacted conditions, which is well known, whether folks cool things, put humidity in, but it's not really by design. So what this plot is actually showing is that if I expose this aerosol, which is bimodal, at 10% RH, the smaller particles basically equilibrate, so they achieve their primary particle droplet size very, very quickly, almost instantaneously. And within about half a second, the larger particles, which are 10% RH, they also equilibrate to their particle size in under half a second. Now, I just want to postulate this, that when someone takes an inhaled dose, from the point it enters your mouth to the back of your throat, it's gone from 55 to 90 plus RH. By the time it traverses your throat, it's almost at 100%. So what's happening on the plot on the right is now the smaller particles, they're equilibrating relatively quick. So even in under 0.2 seconds, they equilibrate quick. But it slowed down a little bit at 100% RH. But interestingly, the larger particle, which are more than likely going to be hanging around the mouth-throat area and the upper respiratory tract. So therefore, either they make it as part of the lung dose or they don't equilibrate within that half a second period. They're taking much longer to equilibrate, which means that they're still changing. So there's time-evolved changes that are occurring in the system. And what we are trying to do now is trying to pin this into, well, how does that impact time-evolved regional deposition? So if I had a time axis on even a simple algebraic model, like the viable lung model, how does that change the distribution? Now, this is not what you build your IVB on. This is not what you go and do your bioequivalence on. This is what you do your product understanding on to better understand how you control your more controlled cascade impaction tests, how to define the humidity, but understand the droplet dynamic and understand how that impacts the behavior. This is a simple system. It hasn't incorporated excipients, hasn't incorporated different salts, which would change some of the behavior of this system. So I just want to conclude by saying, a lot of this wouldn't have been possible without GDUFA funding. I've been very fortunate to have FDA supporting all this work. We've had lots of interesting discussions around this, and it's allowed us to work with the agency to bring some of this types of approaches into more complex aerosol formulations. I think we should, if we're building a Q3 for orally inhaled drug product, it needs to start with PK. It needs to start with showing, if you make formulation changes and you can measure them with these in vitro tools, how does it impact PK? And to build that and to build that into the design principle. So don't just think about, we need to run an IVB on every test. It's build up that orthogonality and pick the tests which you can best control, validate, and can reproducibly conduct to do this. And I think the quality aspects, as folks start putting these types of applications together, I would just say to sponsors, take the time to look at the quality aspects. Really take your time getting the validation done correctly, and that would save a lot of issues when it comes to actually filing. And I think the last point I'll make is these studies should not be just as part of an IVB. These studies should, or these types of approaches, need to grow with the product all the way through its life cycle. So by the time you come to kind of getting into your IVB, you've got the data set to define what needs to go into your IVB program when it comes to having an alternative approach. I'll stop there and just thank all of my collaborators. As you can see, the list of my collaborators from academic and commercial stays the same. The list of the FDA colleagues increases, which is just, it's a good profile because it means that we're spending more time working with the FDA. And I'll thank you for your attention. Thank you.
So first of all, thank you for inviting me here. It's a very long trip from Sydney, but it's definitely been worth it. I'm sort of relatively new to the pharmaceutical aerosol space, sort of got into about 5 years ago. And as I've said to a few people, just yesterday, I've probably learned more than I have in the 5 years of reading papers. So it's been incredibly useful for me. So what I'm going to be talking about today are really laser and optical diagnostics, specifically going to be looking at DPIs because that's what my focus has been on over the last few years. So I come from the perspective of someone who worked for a very long time in spray combustion with specific applications looking at gas turbines, a little bit in IC engines as well. So a lot of my kind of background in terms of how we do what techniques we've decided to apply to dry powder inhaler flows has been from experiences that we've had in that space, very highly dynamic aerosols. In that case, we also have temperature variations, pressure variations, but there are a lot of similarities in many ways between DPIs and the sort of problems that we have there. So what I'll do today is basically go through 3 case studies. The majority of these are, in fact, all of these are a sort of work in progress. So I'm not going to be talking too much about a lot of the published work. I'll point to some of the papers, but that's sort of published. So you can talk to me afterwards and I can tell you where it is. I want to sort of show you some of the sort of very recent things that we've started to work on. And I'll talk a little bit about the motivation behind everything that we sort of do. So ultimately, what we're trying to do and that sort of gap that we're trying to fill here, and Jag has commented on that already, is this sort of dynamic issue. So aerosols are very, very dynamic, and we want to be able to get time-resolved and spatially resolved information on aerosols. So particularly in the context of DPIs, we want to be able to get measurements of what the particles are doing in the actual aerosol in situ, basically, without having to do things like sample it afterwards and sort of influence the aerosol, so to speak. So that's one of the advantages of laser diagnostic techniques. So of course they do have in some circumstances a few limitations. From the context of sort of developing a DPI, we want to be able to come up with methods that can give us quick answers. So something that we can deploy in an existing system that tells us straight away, is the aerosol sort of behaving how I expect it to behave? Is it behaving similarly to this other device that, for instance, if you're developing a generic? So something that can be used to essentially discount ideas very quickly. So a lot of what we do is integrating these sort of high-speed, high-resolution diagnostic techniques into DPI so that we can get this kind of very quick feedback on what's happening. Computational models, we needed 2 sessions. I guess there was too much content, but a big part of what we do is computational model validation. So again, coming from the spray combustion background, we have a series of workshops that run every year where modelers and experimentalists are in the same room and we come up with benchmark test cases. And all the modelers have to model exactly the same configuration, and we learn exactly about the limitations of the model because everyone is modeling the same experiment. It's been incredibly useful. So something like that could work in the DPI space, but I guess that's a point of discussion for later. But a lot of the methods we develop are specifically geared towards CFD models. So in a CFD model, you're solving an equation, conservation equations, where you're essentially resolving how the velocity pressure dynamics of something is changing in a system but a lot of the data that's currently available in this space deals with the final output. So what comes out of the actual oropharynx, what goes into the lungs, but really the CFD model is predicting how something goes from its initial state to its final state, and we're not doing very much to validate that entire cascade of processes. So the point is that we want to be able to focus on that. Newman to understand capsule rotation. So what we've done is we've designed an experiment where we have a motorized capsule, so we're able to get an active control of the capsule rotation, and we can modify the incoming airflow to look at variations in turbulence intensity, flow rates, so we can control everything essentially independently. And that's the experimental setup there on the right. So this is just, I guess, the platform that I'll use to explain how this technique kind of works. So before I show you the microscopic images, just so you get an idea of what this experiment is doing. So these are just the images from a standard high-speed imaging technique where we have the capsule with powder loaded inside it. So we've done this for carrier as well as pure API. So on the left, you have no airflow, and then obviously as you start to increase the airflow, what you'll start to see is more mixing occurring in the chamber, and the particles, as you would expect, flow from left to right. So there's nothing particularly unexpected happening there. This is really just to show you what the experiment is actually doing. But even from this, there's already very basic information that you can extract if you wanted to. For instance, the repeatability of the ejection characteristics from the capsule from one run to the next can be extracted from experiments as simple as these. Now, microscopic imaging looks more like this. So on the bottom right there, that's from an experiment very similar to this one. And on the top, we have a snapshot of what the particles look like. So the idea here is now that you can get this sort of real-time characterization of the particles. You can get the morphology of the particles, you can get the speed of the particles. And we've seen from the two talks before in terms of sort of basic image processing that you can use. So a lot of that applies here as well. The difficulty here is dealing with defocused particles, and we've spent probably the last 8 or 9 years working out how we can deal with that kind of problem. And there's a lot of literature from the fuel spray space in terms of characterizing defocused droplets. And we've sort of just translated a lot of that over into this space. So that gives you an idea of the kind of images that you can extract. And we've published a lot of work on this over the last few years. A couple of those are with the FDA, with Ross, who I guess is in the other room probably. So what can you do with these images? So there's obvious stuff. So you can get size and velocity and qualitative information, but with careful calibration, you can also get things like dose. So for instance, if you look at your actual images, what you can compute from those is some kind of intensity, right? There are various ways of doing that, but you could look at an average intensity per image. You could use a spatially resolved intensity, and you can look at that over time. Now, if you integrate that signal over time, what you'll find is through a calibration, you can actually relate that very closely to the mass fraction of powder. So we did this with mannitol, with a couple of different pure carrier cases from DFE Pharma, various lactohale sort of powders. And essentially what you can get is a very strong correlation between that integrated area and the powder evacuated. So if you do a little bit more work and just look at the integration at different time bands, what you can then get is essentially a mass fraction of the dose evacuated as a function of time. So this is now telling you, as that capsule is rotating, what proportion of the final mass has been ejected from the capsule. So information like this is very useful when it comes to computational modeling. You need to understand dynamically how the particles are actually leaving the capsule. You would be able to use methods like these. The calibration is not particularly onerous, and you don't have to do it for different geometries. You have to mainly do it for different particle types and for different imaging configurations. So it isn't something that is an enormous sort of investment in time. Now, just to give you another idea of some of the kinds of things that we've done with images like these. So this is from published work. We published this very recently, where we're looking at collision dynamics of particles. So something else that we see a lot about in the literature is what's sort of leading to deagglomeration in DPIs. You know, there's particle wall impaction, particle to particle, and turbulence, and so on. And some of that sometimes is a bit hand-wavy. So we have tried to actually directly compute the probability of a collision occurring when a particle collides with a wall, particle colliding with another particle. And again, a lot of that work is just around the image processing. But to give you an example of what we did with this, we determined velocity distributions of particles after conditioning them from colliding apart, from sort of, you've got the colliding and non-colliding particles. What is the velocity, the probability distribution of speed for the particles look like when they collide and when they don't collide? And we developed a really simple logistic model, right? So essentially a statistical model, which was then able to accurately predict the collision likelihood for all of the cases that we ran with 2 different inhaler devices. We just gave it a training set from a couple of different flow rates on one device, and it was able to predict it for the rest. And the disclaimer here is that both of those devices had some very similar mechanisms, right? So they both had a sort of dominant swirl effect and sort of tangential inlets. But it would not be very complicated to expand something like that to apply to other DPIs as well. So I guess the other comment I will make here is that data science has come a very, very long way. And there is actually, we're producing data here in terms of what's happening inside the DPI. There's already a lot more data available and published in the literature. And we are probably, the conditions are probably ripe now to try and make use of sort of even relatively basic modern machine learning methods to try and make certain predictions. But you know, we've done that here. Okay, so I'll move on to case study 2. That's on sort of dynamic particles. And for that particular type of experiment, and we've done that in other types of sort of DPI-like flows as well. The other method I want to talk about now is optical coherence tomography. So if you've been to an optometrist before, you may be familiar with this. If you've had a look at, they've had a look at your optical nerve, OCT is probably what they would have used. So it's not a method that's completely new. It's been around, but it has not really been widely used in particle flow problems, and in fluid mechanics, very, very rarely. So I wanted to kind of talk a little bit about this in the context of deposition measurement. So just very briefly how this works. The crudest explanation without having to look at that diagram is it's kind of like ultrasound, but replace the sound with light. So basically you're shooting light towards a target. That target has multiple interfaces. The light is going to bounce back from multiple interfaces, and that is going to give you some information about the depth resolution. It's really not that straightforward, but that is essentially what it does. More specifically, you've got some sort of broadband light source. You send it down a beam splitter. Half of that light will go to a reference mirror, perfectly reflective. We send that down to a detector. That's our reference arm. The rest of the light, the other 50%, or sometimes we split it so it's actually most of the light goes to the sample rather than to the reference mirror, will reach your sample. Now your sample in this case is a mound of particles or some layer of particles on a surface. There's multiple interfaces there. Because we're using a broadband light source, different wavelengths are going to reflect on different surfaces. And essentially, we can then do a spectral decomposition to work out the location of all of those interfaces based on the wavelengths that we delivered to the target. So that's what's known as spectral domain OCT. There's other types of OCT as well, but they're all essentially kind of doing the same thing. So if you look at the table, it sort of shows you in terms of resolution where it kind of sits. Typically from 1 to 10 microns is the spatial resolution you'd get with OCT, and the depth of field is around 2 to 3 millimeters. That's kind of a really nice sort of area for visualizing layers of particles. Okay, so we published a paper on this a couple of years back, and the experiment that we ran back then was just a really simple DPI that we put into a sort of realistic mouth-throat model, and then we just had a very simplified version of the first sort of 3 bifurcations of a really simple 2-dimensional lower airway. And essentially, what we wanted to do was to see if we can actually visualize these particle layers accurately. And so you get an idea of what this kind of looks like. These are showing you images of the deposited particle layers at a few different locations in the upper airway. And you can also extract information like this from the lower airway. So what you end up seeing is that as the particles land on the airway, they tend to form stacks of agglomerates. So that's even if they're fully aerosolized and then they land on the airway, we don't normally see a monolayer there. We see a stack of particles. So of course, as you go further downstream, if you go really, really deep into the lung, we expect that that would likely become less of a problem. But that is something, again, that becomes quite useful if you want to be able to extract very accurate regional deposition data. That's only one application. OCT can be used for other purposes as well. For instance, if you wanted to look at permeability rate, you can actually do that with OCT. So this is our more recent sort of OCT technique where we've essentially expanded this now so that it provides us more of a kind of tomographic structure of a particle layer. So these are measurements that you do in situ. So the OCT technique can be applied directly to the airway surface or to an aerosol. And what we're doing now is we're making this time resolved. So if that's made time resolved successfully, it means that you can actually get a 3-dimensional map of deposition happening on an airway surface as the aerosol is flowing through that airway. So the nice thing about this is that, as I mentioned, it is depth resolved. So that means that you can look at the particle microstructure at different sort of locations within the particle mount. So the left is showing you 3 micron, or DV50 of 3 micron pure mannitol, and the right is lactohale 100 from DFE Pharma. So you can sort of discern some of the main differences there. There's clearly a lot more agglomerates on the left, and with LH100 you can kind of see some of that, the individual particle structures there. So that gives you an idea of how you can use that. And you can apply sort of relatively standard techniques to try and get some structural information from these images, but it's sort of early days. So we're only still kind of learning how to use that particular type of method. Now, the final case study I wanted to go through are very simple laser extinction methods. So laser extinction or laser attenuation has been used before. It's not anything particularly complex, but I think that it probably could be used a lot more in this sort of area of realistic mouth-throat models. So this is part of a sort of ongoing program we've got with the Proveris Scientific. So we have a number of different projects that we're doing with them. So I can show this one to you, which is part of a PhD project, Zhao Qima. And essentially that's his name. And what we're looking at is trying to understand how we can use a relatively simple setup to track the dynamic movement of an aerosol through an airway. So the way that we've dealt with complex airway geometries in the past, we've published some work looking at sort of PIV and flow fields in airways is because the airway geometry is so complicated, you have to use refractive index matched fluid so that you can see through the airway. Obviously, that means that it becomes completely unrealistic. You can't really deliver an aerosol through it, so you're only looking at the flow. So what we wanted to do is something a little bit more simple, where we've modified some specific sections of the airway so that we can deliver laser diodes through them. And we're basically detecting the aerosol as it crosses through each of those areas. So what you've got on the bottom right is probably not a very useful figure, but essentially that's just the background noise that you're seeing on each of the detectors when there's no aerosol flowing through it. And what you have on the left there is now what the signal looks like when an aerosol flows through the airway. So if you look at the top left, we've sort of normalized that vertical axis, so it goes from 0 to 1. So 1 is maximum voltage, that means you've got nothing there, so the laser is going straight through the airway. And when it goes down to 0, it means it is completely coated, right? So that's how you can interpret those results. So that first graph on the top left is right at the start of the upper airway, close to the mouth. And what we're seeing is a gradual reduction in the intensity as the aerosol flows through, and that's also coating the actual airway wall. When that aerosol has left, the airway wall has been fully coated to the extent that no light is getting through. Now, as you move further down, you'll see that those signals don't go to 0 anymore. Okay, so there's less of those particles that are actually coating the wall because they've landed earlier in the airway. And some regions, that bottom left one, that's just downstream of the epiglottis, where you can see a lot more mixing behavior, right? So what we have now is that difference. It's going from 1 to about 0.85. Okay, so what's happening is because there's a lot of turbulence in that region of the airway, it's sweeping the particles off the airway, and it's keeping that window actually relatively clean. And you can see that because that intensity creeps back up as that aerosol moves away. So essentially in that case, because you have a drop in intensity and it comes back up again, that's indicative of when the aerosol has come and then gone, and it's moved to the next window. So what we're working on now is understanding how we can actually relate that information to a useful quantitative metric. So there are experiments that you can do there where you have to do calibration in a variety of ways to actually relate the local mass fraction to what you're measuring from that laser attenuation experiment. So again, if we go back to CFD, this is actually giving us a real-time dynamic picture of the deposition fraction, or at the very least, the time of flight of the aerosol inside the airway. So that's something that you can measure quite easily from these because it's all synchronized and triggered to the initial breath for whatever inspiratory profile you want. So you can measure exactly how long it's taken for the aerosol to cross from one region to the next. Very simple method, which would enable CFD models to basically actually validate in the region where they're being used. Okay, so that's basically what I wanted to cover. A couple of things in terms of the outlook here. So obviously, I'm biased here, being someone who works in optical and laser diagnostics, but I think there is a lot more potential to see more of these used in the characterization of DPIs. There's a few reasons for that. One, you get that sort of dynamic information, even though in certain situations you may not be getting the resolution that you would like. So with OCT, we get down to about a micron. With high-speed shadowgraph imaging, you can get down to the sort of diffraction limit of the light that you're using. So half a micron is theoretically possible for the pixel resolution, but typically with a pixel resolution of around half a micron, you would only be resolving particles of around 2 microns-ish, right? Depending on the image processing methods that you're using. So there is a limitation there, but it still gives you a lot of very detailed information. I think many of these methods probably need to be a bit more user-friendly. There isn't really a standard in terms of how you're applying these different diagnostic techniques, how you're imaging, what lens you're using, how you're processing them. There are some sort of commercial methods available, but a lot of those are also a little bit different. The final point I'll make that is with CFD. I don't think that CFD is validated appropriately in this field. That's my opinion. I think that a lot more can be done in terms of, and you know, I'm partly to blame to that as well. So we do CFD and we just don't have the data that we would like to have to validate, but we sort of essentially use the results to give us insights anyway. So I do think that we need better experiments with better defined boundary and initial conditions in order to validate the CFD. So that means isolating the mechanisms that we know exist in some of these DPIs and coming up with experiments that closely model those DPIs, and then developing and understanding the limitations of the CFD on that basis. And then you can apply them to more complex problems. That's worked very well for us in the field of spray combustion, for instance. And then understanding with some of these data from the laser diagnostic methods, so obviously particle size and things like that is quite straightforward to understand, but there's a lot more information that you can get out of those images and out of the signal data that we get. Trying to relate that to markers of efficacy of the device, I think, is important as well. Okay, so I'll sort of leave it there. Just in terms of thank you to the research group who's really done a lot of the work that's been presented there. And three of my very close collaborators, Hakim Chan, Xiaokun Cheng, and Dino, who's been involved with various of this work here. And we've been supported, most of the work here has been supported by the ARC, but I'd also like to acknowledge the FDA, who supported some earlier work from a few years ago, where we sort of started looking at some of this particle imaging, and Proveris as well. Okay, thank you.
Good morning. I'm Xian-Ming Zeng. I know I'm between you and a coffee break, so I have my timer set up. So first of all, I would like to thank the FDA to invite me to give a presentation on in vitro characterization studies. You know, instead of talking about individual in vitro characterization studies, I was thinking that we already have enough in vitro characterization technique that can be utilized to support the development of bioequivalent inhalation products. So that's today my presentation. We're focused on how to utilize all the well-defined in vitro techniques to guide the development of bioequivalent products, and how to utilize the technique to support the demonstration of bioequivalence. So of course, you know, the opinions, views expressed in this presentation are solely those of myself and do not represent those of Transparent Bio. Give you a brief outcome of our outlook of presentation. First, I would like to share with you the number of generic inhalers approved by the FDA and the number of hybrid inhalers approved by the UK MHRA. And then, of course, I will briefly touch upon the challenges of weight of evidence approach to demonstrating bioequivalence, as we have heard over the last 2 days repeatedly, that is very challenging to use the weight of evidence approach to demonstrate bioequivalence for inhaled generic products. And you know, of course, I will give a brief overview of alternative approaches proposed by the FDA, and I share some of my industry experiences, ideas on how to design equivalence in the generic inhalers, and some of the possible test methods that are already available to demonstrate equivalence by design approach, and a possible in vitro and PK methods to confirm equivalence on exhibit batches. Basically, you know, if you design equivalence in the generic products to start with, then you reduce the burden on the demonstration of equivalence on a very limited number of exhibit batches, 3 batches. So this is what I would like to present. The first slide shows on the left side, the US generic inhalers on the US market. As we heard over the past 2 days, that there are 4 generics, 4 under generic products for 4 reference products approved in the US. And as you can see on the right-hand side, there are many, many more hybrid inhalers. So I don't want to say generic, I want to call them hybrid inhalers approved in the UK. So this presentation really shows, this slide really shows that the patients in the UK or in the US, in Europe, they have access to much more hybrid or generic inhalers than the patients here in the US. Why? So of course we know that the legal bases are different between the US and the UK, and of course more importantly, the bioequivalence requirement is very different. Of course, in the United States, we know we need equivalent in vitro performance, systemic exposure, local delivery, and Q1, Q2, etc., and it must be interchangeable. In UK and Europe, bioequivalence is considered not being able to demonstrate through a bioavailability study, therefore it's called hybrid product. So there's no need to show interchangeability. And of course, in US, we have a specific guidance, product-specific guidance, and in Europe, they have one guidance on the requirement for clinical documentation. And the approach here in the US, we have aggregate weight of evidence approach, and in UK, Europe, we know they use stepwise approach from in vitro through PK to PD. And the labeling, in the US, we need to have the same labeling with minor or permissible differences. In UK and Europe, labeling can be different, so they are different products. So in the US, generic product is substitutable. In the UK and Europe, hybrid product is not substitutable. So you can say, that's the reason, they actually effectively they're different products. So of course, on the left-hand side is weight of evidence approach, and on the right-hand side is stepwise approach adopted by UK and Europe. With the stepwise approach, you can actually receive approval for a hybrid product based on in vitro comparative data only, as long as you meet all the in vitro criteria. If any of the criteria is not met, then you go to deposition, equivalency in pulmonary deposition, which can be demonstrated by both PK with without charcoal or by imaging study. And we know that if you look at over the past 20 years, and the majority of the drugs are approved, the hybrid products are approved based on PK study only. And the imaging study has not been used to support the approval of any product, as far as I know. And of course, even no DPI has been approved through the in vitro comparative data only. So if you're solution MDI, you may be able to get approval based on in vitro package only. Whereas for DPI, you have to do the PK study, PK with charcoal, as you know, to demonstrate lung deposition, without charcoal to show systemic exposure for safety. And if you are unable to demonstrate bioequivalence based on PK study, then you have to do PD study, and also you have to do full clinical program. So over the last 2 days, I think it has been, acknowledged by many speakers that our weight of evidence approach has put too much weight on the PD and the clinical endpoint studies. And we all know that PD and clinical endpoint studies are costly, time-consuming, and insensitive to demonstrate bioequivalence between two products. So because of that, we know the FDA has actually long expressed interest in alternative approaches. So much so that actually there's a guidance document, as you know, on beclomethasone on BDP. And this guidance document recommends alternative approaches. This is what we are working on here. We're talking about, so basically why BDP? Because according to the guidance document, the BDP aerosol is a solution-based formulation. As long as the test formulation is Q1, Q2 the same as the R formulation, the device sufficiently similar to the R formula device with respect to critical design attributes and the user interface, then we could use these supportive in vitro studies to help ensure equivalent deposition as a local site of action. So I think over the past 2 days, we We heard people talking about more predictive APSD testing, and also all the range of in vitro studies, such as aerospace with respect to velocity, evaporation rates, dissolution, MDRS, and also quantitative methods and modeling, also tentative in vivo PKB studies. So when I was reading this guidance, I was thinking the agency was already thinking about the criticality of design the product, design equivalence into the product. So because, if the product is Q1, Q2 the same, if the formulation is Q1, Q2 the same, if the device is similar, then that means equivalence has already designed into the product. Then the BE testing is just the only confirmatory testing. So then that made me think that, if we took a holistic approach and to design equivalence into generic products, to make sure that the formulation is Q1, Q2 the same, and also Q3 is similar, and the device is similar in terms of critical design attributes, and the product is similar in terms of device user interface, and that means our equivalence is already designed into the product. So our demonstration of equivalence will not only rely on the testing, BE testing of 3 exhibit batches, but more and also rely on the product design. So this kind of approach is actually will be less reliant on final testing, which only demonstrates BE of the 3 batches tested. But it's also with the equivalence designed in the product, we can assure that the product are bioequivalent throughout its entire life cycle. So because of that, I was thinking, can we can still take a weight of evidence approach. The current weight of evidence approach focus on the clinical endpoint study, which we know is not sensitive to differentiate different products. And instead of doing that, can we actually take a weight of evidence approach focusing on the design and development product? So how to do that then? Of course, we can take a similar approach as a quantity by design approach. In order to design equivalence into the proposed generic product, of course, firstly, we have to fully characterize the R&D in terms of the formulation, container closure system, Q1, Q2, Q3, device, usability, performance, etc. We need to characterize multiple R&D batches at different stages of life from the target market and conduct very, very thorough characterization to establish the full profile of the R&D. Then based on the R&D profile, then we can define the quantity targeted product profile, QTPP, for the proposed generic product from a product design, including API, excipients, formulation characteristics of Q3, container closure system for MDI, and a container closure system for DPI. And we'll look at the performance characteristics for DPI, MDI, as well as some critical quantity attributes, the robustness of products, and the usability. So we define the QTPP of the test product, and we cover all the quantity attributes that are critical for the safety, efficacy, and the quality of the test product, and that are critical to ensure that the test product is bioequivalent to the reference product. Then after that, then we can take we can take both a quality by design and a quantity by design approach to develop our generic products, to ensure that our generic product is equivalent and consistently meet the quantity requirement throughout its entire life cycle. So yesterday I saw a very similar slide presented by Dr. Newman. And so, he listed the different steps. And of course, my presentation, my slide, I put together my slide independently. So, these are the different steps of a drug from formulation through actuation, inhalation, delivery, deposition, dissolution, and absorption, and distribution, metabolism, elimination. So the entire process from formulation until elimination from the body. So we can look at each steps, individually, and to say how we can do to make sure our test products are equivalent to the reference product. So we can actually we can look into each criteria and what method we can use for each criteria to ensure that the two products are bioequivalent for each of the steps. So for example, for the MDI, we can look at, of course, Q1, Q2 formulation. We can use MDRS, look at the particle size and shape for suspension formulation. For suspension formulation, we can also look at deagglomeration, sedimentation, or creaming behavior after shaking. We can look at the chemical composition of valve material, canister surface, and also determine X-valve delivery. And for the actuation, of course, we can look at the user interface comparison, force to actuate, etc. And for delivery, we can look at the spray pattern with, without airflow, and droplet size distribution with, without airflow, delivered dose with a spacer, and with one spacer, delivered dose at a different shake delay time interval for suspension aerosol. And of course, when in terms of deposition, we can look at the actuator orifice diameter comparison, because we know actuator orifice is critical for the APSD profile. We can also look at the APSD with, without coordination, APSD with, without spacer, and also realistic APSD profile. I believe with the combination of all the tests, if we can demonstrate the test is comparable, equivalent to the reference product, that means that the equivalence has already been designed into the test product. Likewise, for the DPI, and we can also employ a range of in vitro characterization techniques to analyze the formulation composition Q1, Q2, and also formulation physical chemical characteristics Q3, and we can also analyze delivery characteristics. Particularly, I would like to focus on, for example, for DPI, it's really look at the flow dependence, the different flow rate, and also realistic APSD profile. And for example, for the capsule device, we can look at capsule water content, capsule piercing, peeling, or fragmentation behavior. For blister structure for blister-based device, we can look at the blister structure, saline, peeling, or piercing characteristics, etc. So again, I believe that with the combination of all the methods we have so far, and if we can demonstrate that the test is equivalent to the reference, that means the test has already been designed to be, equivalence has already been designed into the test product. So once we have achieved that, of course, then we need to demonstrate after deposition, then you have dissolution and then an ADME. The dissolution, we know the dissolution test, a lot of work. For example, Gunther's group, have done a lot of very good work for dissolution test, which is very relevant for DPI or for solution suspension. Even could also be relevant for solution aerosol. I remember a few years back, I saw a paper published by Ben Forbes group at King's College London. They compare the PK for Cuva and between Cuva and the cleaner modulite. And the cleaner modulite contain glycerol. So although they're both solution formulation, the particle size could be the same. However, since since the modulite formulation containing glycerol, and if glycerol is a non-volatile excipient, so the evaporation rate is probably slower. So you end up with a more crystalline particle. So the PK profile is very different. So because of the crystallinity of the BDP particle after deposition, which is affected by the formulation differences. So that's why dissolution is very critical, not only for suspension or DPI or formulation, may also be critical for the solution aerosol. And in terms of ADME, of course, we know PK without charcoal has been widely used in the UK and in Europe for demonstrating systemic exposure, of course, has also been used by us here in the US. And we also, PK with charcoal block has been widely used to determine local total lung deposition for efficacy. And we can also use physiologically based PK modeling to calculate the ratios of central to peripheral lung deposition for local deposition. So this may only be applicable to anipophenic drugs. So there are other in vitro methods that could also provide supportive evidence for BE. Just name a few: mass-based plume geometry, mouth rate model. Of course, there are a lot of studies on mouth rate model, simulated inhalation profile. I think the last one, I think this has a huge potential. 3D printed lung models. As somebody mentioned that before. I was thinking, if we have this 3D lung models, which is, can be standardized. And if we can use this 3D lung printed lung model, 3D lung model, couple that with a mouth throat model, coupled with this realistic flow profile, then this, if we can standardize this method, then it could be a very, a representative method for, quantifying lung deposition. So just a thought. So my concluding remark are basically bioequivalence can be designed into generic inhalation product, which is consistent with the FDA's guidance on BDP. So if and equivalence by design can be demonstrated by comparing test reference product with respect to formulation, composition, device, design, a drug device combination, and a patient device interface. So we already have a wealth of technologies that can be utilized to demonstrate that the equivalence has already been designed in the product. For generic product developed by equivalence by design and quantity by design, it may be sufficient to conduct confirmatory BE testing on exhibit batches using in vitro PK studies without the need for conducting clinical endpoint and PD studies. I believe this holistic approach with a focus on the design and development, I will ensure equivalence not only on the exhibit batches that has been tested, but also on future commercial batches throughout the entire life cycle of a proposed generic product. Thank you very much for your attention.